let's just recap on protein function. So cells, there is a difference between prokaryotic cells and eukaryotic cells. The proteins in these two essentially are functioning similar, and what's happening? The eukaryotic cell has some subcellular organization. It has mitochondria, for instance. It has this structure called Golgi apparatus, then the plasmatic reticulum. Most importantly, in some sense, it has the nucleus. Proteins perform functions in different compartments, perform functions that have different sort of overall concepts. DNA maintenance, translation, structural support is a function of a protein. They capture light. There are real motors. They are sitting in the membranes. They're making gates, holes in the membrane. All of that is about protein function. Now, when we look at the numbers, the database of all known sequences has about 81 million sequences. The database of annotations, so there is something known about that function, is called switchpod. That is about 500,000. So this is roughly 2.5 orders smaller. And there is no automated or no reliable automated prediction of protein function from amino acid sequence yet. This is why we have to do a detour. Function itself is a word that is explained on different levels. I talked about the gene ontology. That is essentially an ontology or language describing protein functions on 3 different levels. On the level of cellular component, that essentially where in the cell you are. This is what we are still talking about today. Possibly we are completing the story of cellular component today. Then there's this issue of molecular function, that is essentially how the protein functions, how in detail atoms are bound, how bonds are cut, how they function as enzymes, whether they function as enzymes. And what you see in this video here is that movement, so vibration in this particular case is relevant for function. Without movement, it doesn't happen. And then there's the biological process. So there's one particular protein, HIF, that activates some other proteins. It needs some molecules out there. Under some conditions, it goes this pathway, and other conditions, it goes this pathway. In terms of subcellularization, again, one important issue to make here is that most proteins that are drug targets are extracellular proteins. Why do you believe this is the case? Why are most so drug targets? Now, to repeat, extracellular and membrane proteins make up most of the drug targets. Mislocalization lead to diseases. Here's a set of examples where the reason that the protein being mislocalized, ending up in a non-native localization, leads to a disease. And there are many. This is just an early table here. There are many of these examples. So localization is important, and here's an example where mislocalization leads to a particular disease. It's difficult to see. Let's just move on. You'll have it on your slides anyway for visual. So we want to predict subcellular localization because we are not knowing it for all proteins, because knowing it helps us a little bit to know about function, and essentially we can. So those kinds of arguments, this is a little bit going back to what you said, we do what we can in some sense, right? And ultimately this goes back to the zip code idea. So high throughput methods for subcellular localization do cost. They're not completely accurate, but most importantly, they're not completely complete. And this is an outdated number, but despite a lot of effort in human, we still have, of the 20,000 human proteins, for at least 8,000 localization is not experimentally known, despite 20 years of work. So there's still space for prediction methods, and prediction methods sort of try to use the pathway that exists for transport and use motifs that are sequential, or even in some case motifs that are visible on the structure. These motifs you can sort of think about as address tags or zip codes, and that's the Blobel who got a Nobel Prize for sort of this idea of zip codes. I talked about predicting them through identifying these signals like sorting signals, signal peptides and the like, through homology-based inference, through text-based analysis. And I sort of had some first ideas how to predict de novo from sequence alone, and sort of some hybrid approaches. And today I'm going to go a little bit more into a de novo prediction using machine learning that was developed by Tatjana Goldberg, that sort of mimics the pathway here to predict subcellular localization for eukaryotes, but also for bacteria. Again, in bacteria the story is much simpler because essentially you have inside and outside, but still this can also be predicted. If you want to predict with a novel method, you always have to begin with a data set assembly. And let's begin here. So again, for bacteria and archaea, there are only fewer classes, 6 for bacteria, 3 for archaea. For eukaryotes, we have 18 classes. And in this particular case, we have 1,600 proteins. So 1,600 proteins means, for 1,600 proteins, we know at that time that Tatjana did this slide here, we knew the subcellular localization. Today we know that for more. I showed you in the log-key story a better version of a Swissprot file. In this Swissprot file you essentially have an annotation that you grab out. So what you have to do, you take the proteins of known localization, you take a sequence unique subset, you line them all against all, that gives you a threshold for homology-based inference. That means essentially you have to do redundancy reduction, and when you do a redundancy reduction, you go from something that has a lot of annotations, here that's the green is the eukaryotic number, to something that has very, very few if you look at sequence unique. The 1,600 that I talked about is sequence unique, that's this number. So you start off with a number that is much higher, and then you reduced. Sequence unique means that if you have a sequence redundancy reduced data set, you can sort of take any pair of these proteins in that data set for testing and for training. Okay? So you can randomly, if that is your redundancy reduced set here, this is the annotated full set, that's the redundancy reduced set, so this little one here, that's the big one. And then you can take any slice of this and do a sort of training testing. Now, the problem is that of course 1,600 is a small number. I cannot get new experimental data. So again, we pick some redundant protein in the vicinity of the training proteins, as long as they are not similar to the testing protein. And then we do stratified k-fold cross-validation, the standard, and then we add something to the standard. Okay, that gets us to the log tree method. So data set, the training method, and in this particular case we use SVMs. The SVMs, as I already briefly mentioned, they sort of involve projecting your input data onto a higher dimensional space. In this higher dimensional space you have a linear separation between the data sets. Say there are these star crosses and the minus signs. You want to separate them. Those are in fact 4 different lines that would separate them. What the SVM idea does is first picks the points that are in different classes as a supervised model and that are closest to each other. So the points where you have a minus at a plus sign close to it in terms of this line. And for those you essentially try a separation. This linear separation gives you then sort of the best model for the most difficult to learn points, and that in fact is referred to as a kernel function. There are different kernels that we can use here, different string kernels for sequences, as we have word strings, we have amino acid strings here. Let me step back. So far, when we developed de novo methods for the prediction of subcellular localization, we looked at amino acid composition. Now we are sort of drilling this open, and we're trying to in fact put stretches of sequence into this, okay? And I'll get later to how we define these stretches. So one way could be stretches could be motifs, and we know that motifs like a localization signal are relevant for subcellular localization. So it's just about finding these motifs. So there's a subsequence or string subsequence kernel. So that essentially is a k-mer, as simple as that. You have a simple pattern that is a simple k-mer, where k is the number of consecutive amino acids that you use in this pattern, and then there are sort of additional free parameters. Then there is a mismatch kernel. So this is essentially looking at similarity between the protein that you want to predict and the protein for which you know experimentally what the substrate localization is. And you again predict it based on the similarity of these mismatch k-mers. And then there's a profile kernel, and the profile kernel essentially has the idea of using some additional information. And that additional information ultimately goes back to what you may call probability, that a particular amino acid is observed, a particular letter is observed at a particular position, or if we sort of go on to the underlying model, to the evolutionary conservation. So we're looking, in this profile kernel, we're looking at k-mers essentially that are conserved. Meaning, rather than look at something that is an important motif standing out in a sequence, you align proteins and you ask what type of k-mers are the ones where you see sort of the same k-mer throughout a family. Is that idea clear? Again, if I had a bunch of proteins aligned here, and I would say that here are 5 residues, consecutive residues. Here are 5 consecutive residues that are relatively conserved throughout this alignment. Relatively conserved means essentially all members have a similar sequence there for this 5-mer, right? And so essentially I map all of these conserved k-mers in an alignment. So the way we realize this idea of finding conserved k-mers is essentially as follows. In this particular case, we have a multiple sequence alignment, so we have this query sequence up here, and we have 3 sequences here that are aligned to it. So we assume aligned to it means they have similar structure, they have similar function, and in particular, all 4 here we assume have similar substrate localization. So we assume that if there is anything that is making it relevant for the sequence to be in that particular substrate localization, the same thing should be in all 4 of them. The same feature. That's why we want to sort of look for something that is conserved. Now, in the way I write it down here is I turn this around. So instead of writing the sequence in this direction from the first to the last residue here, I write it down. So the MK, okay, it's down. What this one here is standing for are the 20 possible amino acids. So at every single position here, I can have a certain probability or count for what I see at this column. So in the first column here you see it's always M. So you have a 1 on M and a 0 on all the other 20. In the second column here you see it's 2K and 2M. So you have the 0.5 on the K, on the M. K is just not in my list here, so there's another 0.5 on the K, but in this example I don't show K. And so forth and so on. We do that for every single position in the sequence and we get sort of this matrix. Okay? Then we simply do a sort of log frequency conversion of that matrix. And we get log frequency converted, minus log frequency converted numbers. This essentially helps us deal with this old problem of avoiding to divide by 0. We add some sort of minimal number. And then we sort of have, let's say, a probability that in that particular column, in that particular row, so that particular residue here, you have something that is importantly conserved, yes or no. Okay? Given this yes or no, we essentially want to get a comparison between 2 proteins. The protein that is the query, yes, Ebene?
The previous slides, like you put like M0.5, but M is 2M. Like, is it not 2?
No. M is 1.
No, on the second, like this one, K at K. No, the first one.
This.
No, this one. On the left. On the left.
You mean this K or you mean that K?
Yeah. Yeah, the 0.5.
Yeah.
We have 2n. Why is it not 2 third? Like
Why is it not 2?
2 over 3. Why is it 0.5?
Is 1, 2, 1, 2. So it's 50%.
And we count the query.
Yeah. So essentially we can't, I'm sorry, thank you for asking. So essentially, again, this is the sequence here, and then those are aligned. And essentially, we think about it, so well, I see your point. You could just count what is aligned to it, right? But the first sequence is part of the family, so to speak, right? So it has equal, we assume, in this alignment, we assume that all 4 have equal weight. Although this is the sort of, that is the more important one, because that's the query. But we give the query, we could do that differently. And in fact, it is, I'm not entirely sure whether that is sort of behind your question, but the reality is that putting more weight on the query implicitly is done by the way you do the alignment. So you could do the alignment in a way in which this is optimal for the family, or which this is optimal with respect to the first sequence. But that gets back into the last semester, and that gets beyond it. But in this particular case, really, I assume they are completely equal. We have 4 equal sequences, so each one contributes equally. Okay, so let's get back into this one here. Again, the sequence is written in this direction here. This is my 20 amino acids. And let's just begin with a k-mer that has the length 3. And a conservation threshold of 5, which refers to minus log frequency threshold of smaller than 5. And now let's just assume that I represent my protein by this vector. So this vector is a vector of all possible k-mers where k is equal to 3. So it's a vector with 8,000 components, right? 20 times 20 times 20. And for every single one here, I'm going to have an entry which is, in the simplest instance in which we have this method, exactly a binary vector. This is 1 if you have that k-mer, and 0 otherwise. Okay? And again, this is not if you have that k-mer, but the question really is that k-mer conserved? And in order to sort of look at all the k-mers, we look at all the permutations, we look at all the permutations, and we see for all the permutations what are the conservation thresholds and whether they are smaller than 5 or not. In these particular cases, one CAT, AAT, and CQT are smaller than 5, so they pass the test in the minus log frequency, so they're most conserved. And the CAW does not, so that would turn into a 0, and the other 3 would turn into a 1. Okay? Essentially, we look at all the conserved k-mers in that matrix of 20 times length numbers. And again, it's only 8,000 numbers because we have a binary vector. We count it only once. If cat comes again, AAT comes again, or cat comes again, we're not counting it again. We could do that. And then we have another protein, we do the same thing, and then essentially we do a vector product by sort of multiplying these two vectors. And in this vector product, essentially this is what we put into the SVM. Okay? This is what we learn. So it's a simple way. It's a very simple way to use these motifs, create a vector of a high dimensionality, and essentially sort of condense the vector again by simply mapping it to other vectors as a vector product. When you do that, here is a number for string kernel, for profile kernel, and you see 81. This is higher, so 81. The accuracy now this is for eukaryotes done on 10 classes, so it's a Q10. So we ask what fraction of all the proteins are correctly classified in either of these 10 classes of subcellular localization by a string subsequence kernel, it's just a k-mer kernel, and by this profile kernel, and you see 69 is way smaller than 81. So the profile kernel does way better, is what this one says. This difference here is less for archaea and bacteria, because ultimately you're already much higher here, so there's less room to play, so to speak. And this mismatch kernel, we just never got it to work. Essentially, that never worked for our data set. That's why there is no number, because this problem essentially for the mismatch kernel was just too complex. The simpler ones it could handle, but the more complex one it couldn't. So proper kernel is the clear winner. We essentially now have the next issue that SVM creates a binary answer. We have 10 different classes. How do we map the binary answer to the 10 different classes? And essentially, there are different ways in which you can do that. You already mentioned that. We could essentially do one against all. We can have all kinds of ensembles here. Essentially, you can look for the best possible solution, and the best possible solution, ultimately, I believe. So again, let's just repeat them. We had them on the whiteboard, but let's repeat them. The simplest classifier really has the 10 classes here, and every single class or not class, class or not class, and then you pick the highest as a winner. If you have sort of trees of this, then you first predict whether they are, for instance, secreted or not, and then you go further down in the detail. And again, there are different ways of doing this. Ultimately, the solution we found was the one that is close to the biology, and in fact, it was the best one, where the first decision, in fact, is between going to the secretary pathway or, to the secretary path. No, so the first decision here is whether it's a membrane protein or not. On the right-hand side, you have the membrane proteins. On the left-hand side, you have the non-membrane proteins. And ultimately, it goes back to what you said, Fatos. If you have a cell, and there's the nucleus, and proteins in this nucleus of the cell are they called membrane proteins. So in this scheme, what we found the best solution to be is first you look at all the membrane proteins. So you sort of, first you say it's one, in the first decision here, you classify them as going to both to the right. And then you go further, is it that membrane or that membrane? Or is it that outside without the membrane or inside without the membrane of the nucleus? And that is what I said here. The second best thing is in fact the secretary pathway that goes to extracellular and in between to ER or Golgi. And here, for instance, nuclear, cytoplasmic, and some other compartments. In the end, are some compartments here, like the chloroplasts, that are specific to plants. For archaea, there are 3 classes. This thing is much simpler. And for bacteria, it's still relatively flat, relatively simple. There, in fact, in this particular case, you see that the membrane story is not the first decision here. Here, for bacteria, the first decision is it cytoplasmic or not. So again, I said we sort of, for eukaryotes, copy the biological path. We do it slightly differently for different organisms. And that's back to the eukaryotic plot that I showed you. And this goes back to the way we visualize. Okay, so that's secreted, extracellular. Here are, no, I actually don't want to get into the detailed distinction. So I also don't want to get into that. So now we get into the issue. So one issue that I briefly showed you the stratified cross-validation, and the stratified cross-validation ultimately is good only for choosing the internal parameters. Which sort of, are you looking at the string kernel, are you looking at profile kernel, all of these internal parameters. For that, what I did so far is relevant. Now we get into the issue of estimating how well does the machine work in the future for biologists. And that needs sort of a different data set, a different way of looking at it. And there are different things I can sort of look at. In the simplest instance here, I have this old distinction between precision and recall that typically looks only at the positives. Positives means I have a distinction between subcellular localization and not, so nuclear or not nuclear, and then I essentially have true positives and I have true ones and false ones. So protein is correctly predicted in the nucleus and protein incorrectly predicted not to be in the nucleus. So you have essentially 4 numbers here. The numbers that are correctly predicted to be in the nucleus, the ones that are correctly predicted not to be in the nucleus, and the ones that are falsely predicted to be not in the nucleus, and the ones that are falsely predicted to be in the nucleus. Okay, these 4 numbers. This is what people refer to as the confusion matrix, and typically you ought to sort of present your confusion matrix. The confusion matrix is in particular relevant if you have sort of a higher order problem. Higher order, what I mean is you have 10 classes. And what you would see immediately in this confusion matrix is if, for instance, one particular class would almost never be predicted. You cannot easily see that with precision and recall, only if you look at both numbers. So even in an F1, but in this matrix you would immediately sort of spot classes that would hardly ever be predicted. Why could it happen that some classes are not predicted? Okay, so precision, recall, that gets us to. Oh, thank you very much. Is on. Thank you. That gets us to a totally different story. The totally different story is phrased here as a question. When you report performance in a table, and in your table you measure that your device in a 10-class system is 51.52316789. 0. How many digits do you quote?